Major histocompatibility complex, or MHC molecules, are membrane proteins involved in adaptive immunity, which have the ability to display peptides of varying lengths for recognition by T cells. On a healthy cell, the displayed peptides are typically self-peptides, but when a cell is infected, foreign peptides will be displayed. There are two classes of MHC molecules, Class 1 and Class 2. While their basic function is similar, the two classes differ in a number of aspects. Each class of MHC is presented on a different type of cell and in turn are bound by and activate other separate cell types. In addition, the sources of their displayed peptides originate and develop through different processes. Let's start with MHC Class 1, which bind to CD8 positive cytotoxic T cells and receive their peptides from proteins in the cytoplasm.
In healthy cells, there is a constant flux of proteins as new ones are created and damaged ones are destroyed and recycled. This process occurs by the way of ubiquination, which tags peptides for breakdown. Viral peptides are also tagged in this way when present in the cell. These peptides can arrive in the cell by the escape of the microbe from an endosome, or as new viral proteins are synthesized in the cell. You may find it helpful to think of the cells as a hospital in a zombie-filled world, where the walls work to keep out infected invaders. The halls are constantly patrolled, and if a zombie is spotted inside, workers are trained to shoot a tracker into its skin. Protease complexes, which cut the protein into chains small enough to be displayed by the MHC molecule, are also present in the cytosol. They are guided to their peptide targets by the tags made by ubiquination. We return to our zombie, roaming the halls of the hospital, tagged by a tracker. This tracker guides a remote-operated wood chipper to the zombie to break up the creature for transport to a lab.
Production of the MHC signaling molecule takes a number of cellular components, proteins, and molecules. Within the lab of our hospital, the endoplasmic reticulum, exists the cast necessary for such a job. Central to this process is the MHC Class 1 heavy or alpha chain. Though at this point it is little more than a string of proteins, it will become the central aspect of the molecule, the pole, if you will, on which our antigen flag will be flown, alerting the outside world to the zombie danger. Chaperone protein calnexin assists in folding the early MHC molecule. It is joined in supporting the MHC chain by beta-2 microglobulin, which stabilizes and later is necessary for cell surface expression of MHC class 1 and stability of the peptide binding groove. Beta-2 microglobulin is the rope which keeps tension on our flag. After stabilization, the peptide loading complex is formed and calnexin gives way to calreticulin in supporting the MHC chain and keeping it within the ER. Further folding assistance is provided by enzyme ERP57, while the protein tapesin mediates the association between the nascent class 1 molecule and binds it to the TAP transporter.
These peptide fragments migrate to the ER, where they are actively pumped through the membrane by the action of the TAP transporter molecule mentioned earlier. Tapesin allows for close spatial orientation of the MHC1 molecule with TAP, and so increases the likelihood of binding. Some specific immunodominant peptides fit into specialized pockets in the peptide binding cleft of the MHC1 molecule. These specific locations on the peptide are referred to as anchor residues and increase the binding strength between the two molecules. Once again, our poor, chopped up zombie can be used to understand the flow of this progression. The pieces of our zombie are transported into a lab set up in the old emergency room of the hospital. With a controlled environment necessary for lab work, the parts are taken through a set of double doors that require power to open.
Once a class 1 molecule has been loaded with a peptide, the complex is stable and released from association with TAP. It is then exported to the Golgi and transported to the cell surface via an exocytic vesicle, which will present the peptide class 1 complex on the cell surface. Now fully assembled, our MHC has taken on a form similar to a flag flying a zombie limb, used to alert the outside world that the hospital is zombie-ridden.
Now that the MHC class 1 molecule and peptide are presented on the cell, it is functional for recognition by CD-positive cytotoxic T cells. When a peptide is displayed that a cytotoxic T cell recognizes, an immunological synapse is formed. This synapse includes the binding of the peptide, co-receptors, and signal transduction molecules binding to their appropriate counterparts. This sets off a cascade of T cell proliferation and further activation of the immune response. Now let's move on to the antigen presentation process for MHC class 2. These molecules are only found on a few cell types, including dendritic cells, macrophages, and B cells. The proteins that are displayed on class 2 molecules generally come from the extracellular environment and have been internalized by one of the 3 antigen-presenting cells previously mentioned. MHC class 2 molecules are important players in both B and T cell activation.
Once these extracellular proteins are ingested, they must be prepared similarly to those previously used for class 1 MHC molecules, though the method and location of this preparation is different. The protein is brought into the cell with a membrane-bound endosome, which is later fused with lysosomes filled with proteolytic enzymes and acidic compounds. These enzymes break the protein into small peptide chains for the MHC2 molecule. As with before, only a few of these will bind strongly and be immunodominant. Remember our hospital from earlier? Well, this will be similar to capturing a zombie circling the edges of the building, outside of the protective walls. The door of a cage could be left open so that when the zombie wandered in, the cage doors could be closed and the zombie taken inside. This cage will be rolled through the halls to a vat of toxic hospital waste that breaks the zombie apart.
The 2 chains which make up the MHC class 2 molecule assemble in the endoplasmic reticulum, alongside a 3rd chain, the invariant chain, CD74. A portion of the invariant chain binds to the peptide binding groove of the MHC class 2 molecule and prevents peptides, or unfolded proteins, within the ER from binding to emergent MHC class 2. The invariant chain also guides the transport of class 2 molecule out of the ER and through the Golgi apparatus into a vesicle, which becomes part of the endocytic pathway. The pathway is also responsible for the entrance of pathogens and foreign proteins into the cell. Within this vesicle, acidification activates proteases which cleave the invariant chain in 2 pieces. A small peptide, class 2 invariant chain peptide, CLIP, remains bound to the class 2 molecule. The engulfed pathogens, or their proteins, cannot bind to the class 2 molecule due to the presence of CLIP within the peptide binding groove. Specialist protein MHC class 2-like molecule HLA-DM removes the CLIP from the peptide binding groove. It coordinates the CLIP release and the pathogen-derived peptide binding. If we again compare the MHC molecule to a flagpole, we have a construction designed for a new set of displays. The zombie, previously broken down by acid, is that display.
With the peptide in the binding cleft of the stable, mature MHC class 2 molecule, it is ready for presentation. It travels in an endosome to the cell surface where it then fuses for presentation. CD4 positive helper T cells are now able to bind to the class 2 molecule and peptide to form an immunological synapse. If a synapse is formed between a dendritic cell and helper T cell, the helper T cell is activated. If the synapse is formed between a B cell and helper T cell, the B cell has received its second signal and can begin proliferation and class switching. Our hospital is again displaying a flag calling for help, which is recognized and responded to. Both MHC Class I and Class II molecules are the key players in alerting the body when there is an invader. When the body is healthy, the job of MHC is to present self-peptides to prevent the cells from being improperly destroyed. If the body is breached, the MHC molecules will present non-self peptides to alert the army of T cells and B cells. MHC molecules are the vital messengers in the initiation of the adaptive immune system.